Good morning, ma'am. My name is Ayushi Suryavanshi, roll number 140, group 75. So my topic is glycolysis. So first we'll discuss about what is glycolysis. So coming to the next slide. The process of glycolysis occur in all organisms, from unicellular organisms like bacteria and yeast to the multicellular organisms like plants, animals, and human beings. So this process occurs freely in anaerobic environment within cytoplasm, and it is not associated within any organelle. If we observe that glycolysis are the merger of two Greek words, here glykis means sweet, or it can also mean glucose, and lysis means breakdown or splitting. So here from the whole word we can understand that it means breakdown of the glucose. So the process in which glucose of 6 carbon is degraded or breakdowns into molecules of pyruvic acid through the multiple enzyme dependent reactions. So glycolysis is a sequence of 10 enzyme catalyzed reactions that convert glucose into pyruvate with the simultaneous group on the ATP. And the major pathways of glucose metabolism occurs in the cytoplasm of the cell. It also includes the formation of lactate from pyruvate. Coming to the next slide. The major pathways of the glucose utilizations are, here in the diagram we can see that the glucose store the monosaccharides like glycogen, sucrose, and glucose, when glucose breaks down in the presence of oxygen, that is oxygen, and the glycolysis process is performed, it converts it into pyruvate. And in the presence of oxidation with pentose phosphate pathway, it gives ribose 5-phosphate. So the phases of glycolysis leads to the breakdown of 6 carbon glucose into molecules of 3 carbon pyruvate within the enzyme catalyzed reaction, being bifurcated or categorized into 2 phases. So here it means that the glucose consists of 6 carbons, and after the glycolysis is performed, it breaks down into the 2 molecule of pyruvates of 3 carbons, and with the enzyme catalyzed reactions, and the phases of glycolysis are divided into 2 phases. First phase 1 is preparatory phase, and second phase is payoff phase. So coming to the next slide, we'll discuss about preparatory phase. So it consists of 1 to 5 steps of glycolysis in which the glucose is enzymically phosphorylated by ATP to yield fructose 1,6 biphosphate. So here it means here we are trying to say that it consists of 4 steps in which the glucose is converted to the fructose 1,6 biphosphate, and this cleavage requires an investment of 2 ATP molecules to active the glucose mole and prepare for its cleavage into the 3 carbon compound. So here throughout the process we use 2 ATP molecules only, means we utilize only 2 ATP molecules. So coming to the next slide. We'll study about the first phase, that is preparatory phase. So in the first step of the glycolysis, glucose is converted into glucose 6-phosphate by the transfer of phosphate group from ATP to number 6 carbon of glucose. So here we can see that the glucose is converted into glucose 6-phosphate by the transfer of phosphate group from ATP and converted into ADP. So this is the first step, and we got glucose 6-phosphate. After that, and it is not interconvertible, like it is not reversible. So glucose 6-phosphate, now in the second step, glucose 6-phosphate is converted into fructose 6-phosphate by the catalysis of enzyme. Means here the glucose is now glucose. We have written the isomer of the glucose here with the help of catalysis of enzyme. So in place of glucose, we write fructose 6-phosphate. It is converted into the isomer of this glucose 6-phosphate, that is fructose 6-phosphate. In the third step of glycolysis, fructose 6-phosphate is converted into fructose 1,6 biphosphate by the transfer of phosphate group from ATP to number 1 carbon of fructose 6-phosphate. So here we can see that there is a transfer of phosphate group from ATP to the number 1 carbon of the fructose 6-phosphate. So it forms fructose 1,6 biphosphate or bisphosphate. Therefore ATP, and here again we have utilized one molecule, one ATP molecule, and it is converted into ADP. Means second priming reaction. Now after this, and it is not reversible. In the fourth step, glycolysis, splitting of fructose 1,6 biphosphate into two molecules. One is called phosphoglyceraldehyde and other is DHAP. Both are interconvertible. So we are trying to say that after fructose 1,6 biphosphate, we have split it into two molecules, that is phosphoglyceraldehyde and other is dihydroacetone phosphate. So here we can see that the cleavage of 6 carbon sugar phosphate to the 3 carbon sugar phosphate is taking place. So this is the whole process of and of the preparatory phase, and these are the enzymes that are used. First is hexokinase, and second is hexose isomerase and phosphofructose kinase 1 aldolase triose phosphate isomerase. These are the enzymes that we have used in this process. Coming to the payoff phase, this phase constitutes the last reaction of glycolysis, and here four molecules. So here 4 moles of ADP are phosphorylated to ATP, and although 4 moles of ATP are formed, the net result is only 2 moles of ATP per molecule of glucose oxidized, since 2 moles of ATP are utilized in phase 1. So in the payoff phase, we'll talk. It will consist of 5 reactions of glycolysis. Moving to next payoff phases. So the step 5, in this step of glycolysis, an enzyme of dehydrogenase and coenzymes nicotinamide dinucleotide NAD+ work together. They will work together, and the enzyme strips off the 2 hydrogen atom from glyceraldehyde 3-phosphate and phosphate, which are captured by NAD. As a result, glyceraldehyde 3-phosphate will converted into 3-phosphoglycerate. Now PGA picks up the phosphate group and present in the cytoplasm and becomes 1,3 biphosphate phosphoglycerate. So here we have we had 1,3 biphosphoglycerate. Here we have already seen that what happened. The 2-phosphate group is transferred from the NAD and converted into 2-NADH. Okay, so after that we got 1,3 bis or biphosphate phosphoglycerate. Coming to the next step, in this step of glycolysis, 1,3 biphosphoglycerate loses its phosphate group to ATP. Now here this compound 1,3 biphosphoglycerate loses the phosphate group to ADP. As a result, 2 molecules of ATP are formed, and as a result 2 ATP of molecules are formed here. So, and it gives 3-phosphoglycerate. Therefore, it gives a 3-phosphoglycerate here, and this non-reversible. Coming to the next step, in this step, the 3-phosphoglycerate is converted into phosphoenol pyruvate with removal of water molecule. So here we get 2-phosphoglycerate as it loses 1 phosphate group, and after that it loses 2 molecules of water. After that, when it loses 2 molecules of water molecule, it forms phosphoenol pyruvate. So in this step, in the second last step, we have got phosphoenol pyruvate. So in the final step, phosphate group is transferred from phosphoenol pyruvate to ADP. As a result, the molecule of ATP are formed and pyruvate acid is also formed. So here we can see that the pyruvate is formed.